Continuing on with the urinary system in chapter 26. So as we just finished up talking about urine, the filtration step as we're moving through that Bowman's capsule. Of course, what drives all that is the filtration pressure moving from a high to low. So again, our pressure gradients responsible for that filtration forces the fluid from the capillaries across the filtration membrane and into the glomerular capsule. So that a few different pressures here that we talk about is the glomerular capillary pressure. So that's the pressure inside the capillary. Again, it tends to move fluid out of the capillary into Bowman's capsule. Then the capsule hydrostatic pressure. This is the pressure of the filtrate that's already there. So we have the pressure of the fluid coming into it, as well as the pressure of the fluid already inside Bowman's capsule. So before glomerular capsule capillary pressure, what's already in there is the capsule hydrostatic pressure. Then we have the blood colloid osmotic pressure. And osmosis, we're talking about the movement of water. So that osmotic pressure is caused by the proteins in the blood. So remember, most of the stuff can come out into the glomerulus or into the Bowman's capsule, but a lot of the bigger proteins in the cells stay in the blood. So this favors the movement into the capillary from the lumen, so going from a hypotonic to hypertonic. So that osmotic pressure is greater at the end of the capillary than at the beginning, and this is going to cause fluid to leave the capillary and move into the lumen. So between the glomerular capillary pressure and this colloid osmotic pressure, there's a driving force of the fluid in the filtrate from the capillary into the capsule. And that filtration pressure is determined by the glomerular capillary pressure minus the hydrostatic pressure minus that colloid osmotic pressure. So that osmotic pressure in Bowman's capsule is about 0 inside of there. So again, it's causing everything to move. So during diseases, though, like glomerular nephritis, which is infection or inflammation of the nephron, protein actually enters the filtrate. Again, we mentioned in the last one, there's typically very little protein in the urine. But in the glomerular nephritis, there is protein in the urine and in that filtrate. So it exerts osmotic pressure that increases the volume of the filtrate. So it would make it more than it normally would be. So that high glomerular pressure results from low resistance to blood flow in the afferent arterioles, low resistance in the glomerular capillaries, and then a high resistance in the efferent because of that small diameter. So that filtrate is forced across the filtration membrane, and the fluid moves in to those paratubular capillaries in the Bowman's capsule. And then hypertension, which is high blood pressure, can damage those capillaries by creating too much pressure that actually damages those. So looking at the pressures going on here, again, the main pressure is that glomerular capillary pressure that is causing things to come out of those capillaries and into the filtrates, so moving into the convoluted tubules. Pressures opposing that are the capsular pressure and that osmotic pressure, but they're not great enough to overcome that glomerular capillary pressure. So that's why the overall pressure, you take the glomerular minus the capsular pressure minus the colloid to get your overall net filtration pressure. As far as regulation of these rates, there's auto-regulation, just meaning it's intrinsic. So this involves changing of the constriction of those afferent arterioles, which would then change the pressure. So there's a myogenic mechanism. So as the main blood pressure, so systemic all throughout the body, blood pressure, if it increases, then the afferent arterioles constrict, and that's going to prevent an increase in renal blood flow. And then there's tubuloglomerular feedback. So an increased rate in blood flow of that filtrate past cells of the macula densa, that signals is sent to the juxtaglomerular cells, and that causes the afferent arterioles to constrict. And then again, constriction of those is going to help regulate that filtration rate. Now, the extrinsic mechanism, so we have autoregulation, intrinsic, we also have extrinsic. So this is controlled by the sympathetic, again, fight or flight response, as well as hormones. So during severe conditions like a hemorrhage or dehydration, this is when these would become a factor. What's going to happen is the sympathetic simulates causes constriction of those arteries and arterioles, again, decreasing renal blood flow formation. Because again, if we're running and fighting, we don't want to be urinating as we do so. So we're going to be decreasing that urine infiltrate formation. As far as hormones, renin gets secreted by those juxtaglomerular cells. We mentioned those in the last video. So this is going to form angiotensin, which stimulates constriction of those arterioles and maintains that resistance. So again, we have intrinsic as well as extrinsic that are ultimately changing or constricting those afferent arterioles. Now, if we switch over to our tubular reabsorption, again, this is that transport of the water in the solutes from the filtrate back into the blood. So it's just occurs as it flows through the lumen of that renal tubule. Substances get transported into the interstitial fluid. And again, this is going to be that reabsorption into the peritubular capillaries, and it includes like sodium, potassium, calcium, as well as bicarbonate and chloride, and about 99% of the filtrate actually gets reabsorbed. So again, if we think about all the blood that flows through our kidneys, we're only excreting about 1 to 2 liters. So the majority of what's being filtered out gets reabsorbed. So some portions that are not reabsorbed, that's going to actually be what ends up in the urine. And that includes things like urea, uric acid that comes from protein, breakdown, creatinine, potassium, and a few other substances as well. All right, now moving into the proximal convoluted tubule, this is where the majority of that reabsorption is going to occur. So remember, the cuboidal cells here in the proximal have more microvilli for that increased surface area than those on the distal tubule. And again, this is because most reabsorption is happening here in the proximal end. So each cell here, each of those cuboidal cells have an apical membrane that's attached to the renal tubules, the basal membrane, which is on the inside wall of the renal tubules. Again, that's where the microvilli would be sticking out, and then the lateral surfaces that are between the cells. So there's active transport, again, meaning that we need to have some type of energy to move sodium across the basal membrane to the cytoplasm, into that interstitial fluid. That's linked to reabsorption of most solutes. So again, sodium comes in, that active transport then provides energy to allow for reabsorption of those solutes. So there's that kind of payoff. Energy debt is paid by sodium. So because of that active transport, again, sodium is kept low inside this cell. So as sodium moves into the cell through the apical membrane, other substances move by symport, just meaning in the same direction into the cell and retained. So again, there's active transport similar to our neurons, where the sodium-potassium pump puts sodium out. So sodium comes in, it causes an action potential. It's just instead of generating an action potential, here in the convoluted tubule, the sodium comes in as it moves with its concentration gradient. It provides energy to move other molecules to allow for reabsorption. So again, that active transport, like the sodium potassium pump, is important to keep that concentration where it needs to be so that the influx of sodium releases energy. So how many of those carrier molecules or transport proteins, that's what's going to limit the rate. So in diabetes, the concentration of glucose exceeds the transport. So there's more glucose in the blood than there are transporters to move it. So it's not that it can't move. Again, if you think about them, again, as being, the transport protein as being tunnels, if there are only a couple tunnels, there's only so much that can move across the membrane at a time. Whereas if there were, 50 tunnels, more of it could move. So with diabetes, when glucose concentrations are high, that means most of it is not going to get reabsorbed because there's just not enough transporters to keep up with it. So the glucose in the plasma, again, stays in the filtrate and it gets expressed in the urine, which is why doing the urine test and having high glucose indicates a problem. Now, there is some diffusion between cells. So from the lumen of the nephron to the interstitial fluid, and that rate, again, of the transport is the same solutes through the cell. So this includes potassium, calcium, and magnesium. So it just depends on how quickly they're coming in. That determines the rate. And then that volume is reduced by about 65% due to the movement of water or osmosis of water. All right, so if we look at this reabsorption, so again, here's the lumen of our tubule that has the filtrate coming in. Here's our apical membrane, has all these microvilli, and then our basal membrane that goes into the interstitial fluid. So again, here's our filtrate coming through, and then reabsorption back into the capillaries. So that's happening at the basal membrane. So this interstitial fluid would be then going into the capillaries. All right, so our active transport here, again, that sodium potassium pump, just like we saw with neurons, that's going to use energy to take sodium out of the cell. That's going to keep the concentration inside really low, so that then on the apical membrane, now as sodium comes in to the cells, that releases energy because it's going from high to low. That energy is then used to move chloride ions, glucose, and some amino acids into the cell. So it's providing that transport for them using sodium for energy, as well as osmosis that occurs across that membrane that moves water in. Then on our other side, that reabsorption, again, we get Symport here with the potassium and chloride both going out because, again, potassium now is high due to our sodium potassium pump. And then just facilitated diffusion. So then going from a high to low concentration, we get facilitated diffusion, removing those potassium, glucose, and amino acids that we actively transported in from the apical side. They get reabsorbed by facilitated diffusion into the capillaries. Same thing with water moves then by osmosis back into those capillaries. And I just went ahead and left these in here. Again, these couple slides just in a lot more words talk about what I just mentioned, so I'm not going to reread them to you. But again, just pointing them out here in the PowerPoint to help you read through it and understand the steps if you didn't quite get everything that I just mentioned in the diagram. So we get the reabsorption, again, our sodium potassium pump. Again, then we get those carrier proteins on the apical membrane using the sodium to help move them in. Again, on the on then our basal side, then we can get facilitated diffusion, removing those molecules for reabsorption into the interstitial space. And now we're at that nephron where we get some reabsorption here as well. So in the descending into the medulla, again, some of the nephrons go deep, others not so deep. But again, our interstitial fluid has a lot of solutes in there, so that descending segment is thin, again, very permeable to water, urea, sodium, as well as other ions through passive diffusion or facilitated diffusion. So we don't need energy for that. The water moves out of the nephron, so some solutes are coming in the volumes, reducing it by about another 15% added on to the 65% that we just mentioned. So then the ascending thin segment isn't quite as permeable to water, so most of the water is reabsorbed before we get to that ascending limb. But the solutes are still permeable, so there are still solutes moving in the ascending limb back into the into that interstitial space, and those solutes diffuse into that descending vasa recta. All right, so then in the reabsorption in our loops, so the wall of the ascending limb, again, is not very permeable to water. Sodium, though, is going to move across the wall by active transport, again, at sodium potassium, to set up the gradient so we can move potassium and chloride by symport, using sodium going with its gradient to move those against their gradient. So then at the end of the loop, inside the nephron, just looking here at some numbers, the 100 milliosmotic per kilogram, and then our interstitial in the cortex is 300. So then that filtrate within the distal convoluted tubule is much more dilute than the interstitial fluid that surrounds it. All right, so again, if we look here at this descending limb, where we have those squamous instead of the cuboidal down at the end of the descending limb, we now have squamous, which are very permeable to water. So this is where we're going to get that reabsorption of water through osmosis occurring across those simple squamous into the interstitial space, and then ultimately into the ascending vasa recta, back into the capillaries and into our blood flow. Now, as we start to make the bend in our descending loop, we still have the simple squamous, but they're becoming less permeable to water at this point, but still very permeable to solute diffusion. So we still have molecules and solutes moving across in that in the actual loop of our descending loop. Then as we move into the thick segment of the ascending, now we're back to those cuboidal cells that you can see here. And so here we have, again, very little water, so not a lot of permeability. So at this point, there's no osmosis of water, but there's still sodium that is moving by active transport that allows that co-transport of the potassium and the chloride. So there's still some reabsorption happening of those ions, just not permeable to water at this point. And again, this is just kind of going through the actual words and all of that. I'm not going to read it to you. It's what we just kind of described in the pictures. But again, left these in the PowerPoint if that helps you to read through it more so than listening to it. Now, reabsorption in the distal convoluted tubule and then into the collecting duct. So again, water moving by osmosis from the distal tubule to the collecting duct. That's based on the influence of the antidiuretic hormone. So the permeability of the wall is very permeable to water. Potassium and hydrogen ions are not absorbed until further in the distal convoluted tubule, even collecting ducts under hormonal control, depending on the body conditions. And then the urine can vary in concentrations from low volume to high concentrations to high volume and low concentration. So again, very concentrated urine to very dilute urine, depending on the conditions of the body. So what changes those concentrations of urea and other solutes? So urea enters that glomerular filtrate. Again, urea is from the the removal of the amino group from our amino acids as amino acids and proteins are broken down by the body. So as the volume of the filtrate decreases, so again, less water, now it's going to concentrate the urea and it becomes more concentrated. So then the walls of the nephron are not very permeable to urea, so we're not really reabsorbing that. So only about 40 to 60% gets reabsorbed. So there's a significant amount in the urine. Urate ions, some creatinine, sulfates, phosphates, and nitrates are partially reabsorbed, but again, their concentration in the urine is pretty high, not a lot of reabsorption. And then the toxic substances would not be reabsorbed. They're going to be eliminated. And then our tubular secretion as we round out our urine production. So tubular secretion, again, is the movement of non-filtered substances from the blood to the filtrate. So again, it wasn't filtered out of the blood, but now it's going to be secreted into the filtrate. So these, again, are things from metabolic byproducts. So during liver and metabolism, drugs are secreted, as well as some other molecules not normally made by the body. And this can be active or passive. Some of them include like ammonia. This is produced by our epithelial cells during the deamination of amino acids. So again, most of the time it gets converted to urea, but sometimes is ammonia. So that would get secreted by diffusion. Hydrogen ions, potassium, penicillin. So again, thinking about drugs. And substances like aminohippuric acid, PAH. These are actually actively secreted into the nephron. So again, using energy to pump them out of the body. So this neuronal pathway for olfaction, so hydrogen ions get secreted into the filtrate by what's called countertransport in the proximal tubule. So what happens here is hydrogen ions either diffuse from the peritubular capillaries into the interstitial space, then to the epithelial cells, or they're derived from reactions between carbon dioxide and water that makes carbonic acid in those cells. And then sodium and bicarbonate are co-transported across the basal membrane and then diffuse into the peritubular capillaries. And then our hydrogen and potassium, ions are secreted, again, released into the filtrate by countertransport, meaning sodium and potassium move by active transport, and then sodium and bicarbonate co-transport into the interstitial space and then diffuse into the peritubular capillaries. So again, just showing that secretion of hydrogen and potassium into the tubules. Okay, so again, here's our interstitial space. Here's the filtrate. But again, these molecules weren't filtrated, but what's happening here is as sodium comes into the cell, that's going to take hydrogen ions out. So instead of being symports, they're going in opposite directions. And those hydrogen ions, again, can come from carbon dioxide and water, making carbonic acid that then breaks down to hydrogen ions and bicarbonate. So the bicarbonate gets reabsorbed. It goes back into the interstitial space, but the hydrogen ions are secreted. And then again, our sodium potassium pump here is going to keep our sodium concentrations high outside and low inside to provide energy for all of the transport to occur. Okay, and potassium happens in a similar way. So again, this is our filtrate. So the sodium, potassium keeps sodium low inside. So as sodium comes in here from the filtrate, that provides energy to move potassium out and secrete it into the filtrate to allow for that secretion of the potassium ions.